Good morning, everyone. Today I'm going to explain you the topic immunology. So, in today's class, we are going to see about the introduction regarding immunology and what are the cells involved in the immune system and what are the different types of immunity we are having. So, under different types, we are going to discuss about innate immunity and adaptive immunity. So, before starting all these things, I just want to tell you what is antigen, what is antibody. So what is antigen means it is immune response developed against pathogen and wide variety of agents like chemicals, biological products of plants, animal origin. Such agents which will produce the immune response will call it as antigens. So the agents against the body produce an immune response that agents will call it as antigens, whereas these antigens will bind with the antibodies or T cells. Coming to the antibodies. Antibodies are nothing but these are the glycoproteins which protect us from the microbial infection by binding with an antigen. These are also known as immunoglobulins. Next, coming to immunity. So what is immunity? The ability of a body to protect against the type of foreign particles uh, which enters the body, we'll call it as immunity. So what are the cells involved in the immune system? So under introduction, we have discussed about the antigen, antibody and what is immunity. Coming to the cells of the immune system. So there are different types of cells. First one is leukocytes, next is lymphocytes, dendritic cells, antigen presenting cells and macrophages. I'm repeating again, leukocytes, lymphocytes, dendritic cells, antigen presenting cells and macrophages. So we'll be seeing one by one. So what are these leukocytes and what is the role of these leukocytes in the immunity? So what are the different types of leukocytes we are having means neutrophils, eosinophils, basophils, monocytes and macrophages, lymphocytes. So neutrophils helps in phagocytosis of pyogenic microorganisms. Coming to eosinophils, they play an important role in the parasite phagocytosis. Coming to the basophils, they played an important role in delayed hypersensitivity. Monocytes and macrophages helps in phagocytosis, whereas lymphocytes plays an important role in acquired immune response. Coming to the lymphocytes. Generally, the lymphocytes are formed from the hemopoietic stem cells. These hemopoietic stem cells gives rise to lymphoid stem cells, which is divided into small lymphocytes, large granular lymphocytes. The NK cells or null cells are coming from the large granular lymphocytes, whereas the T lymphocytes and B lymphocytes are coming from the small lymphocytes. T lymphocytes again divided into T helper cells, T cytotoxic cells, and T regulatory cells. So, finally, the lymphocytes are divided into T helper cells, T cytotoxic cells, T regulatory cells. Coming to the B lymphocytes. The B lymphocytes first when the antigen get attached, it will be activated, then it undergoes proliferation and then differentiated into plasma cells and memory cells. Plasma cells will produce the antibodies, whereas the memory cells helps in producing the lifelong memory to the body. Coming to the T lymphocytes. T lymphocytes plays an important role in both cellular and humoral immunity. There are different T lymphocytes as we discussed in earlier slide. They are T helper cells, T cytotoxic and T regulatory cells. T helper cells, it will have CD4 molecules on their surface and it plays an important role in both cellular and cellular and humoral immunity. Whereas cytotoxic T cells will have CD8 molecules on their surface. It plays an important role in cellular immunity. If we see about the helper T cells, they play important role in both cellular and humoral, whereas cytotoxic T cells plays an important role in cellular immunity. Coming to the regulatory T cells, these cells will have CD4 and CD25 molecules on their surface and it will suppress. So we have to remember this thing. We have to suppress, these regulatory T cells will suppress both cellular and humoral immunity. Coming to the natural killer cells are null cells. So by the presence of some cytoplasmic granular content, they are capable of killing the microorganisms or any tumor cells or virus which are present in our body. They act by themselves, naturally act as killer cells. They will have CD16 and 56 on their surface. Coming to the antigen presenting cells. So antigen name itself says antigen presenting cells. So these are the cells which will capture the antigen and present to the surface after binding with the MHC molecules for recognition by T cells. These are essentially once the antigen get exposed, then only the T cells will be activated. Coming to the dendritic cells. Dendritic cells resembles the dendrite of nerve cells. They are present in the blood epithelia in all the organs. So there are different dendritic cells. If we see at different organs, skin and mucous membrane will have Langerhans cells. Blood and lymph will have circulating dendritic cells. Organs like heart, lung, liver, kidney will have interstitial dendritic cells. Thymus will have interdistating dendritic cells. Lymph nodes will have follicular dendritic cells. So these are the different dendritic cells which are present at different organs. Coming to the macrophages, these are coming from the circulating monocytes. So once they stay for 8 hours in the blood, they will migrate into various tissues and then they differentiate it into the macrophages. So this is about macrophages. So we are having different macrophages again at different places. Lung, we are having alveolar macrophage. Histiocytes is present in the connective tissue. In the liver, we are having Kupffer macrophages. Kidney, mesangial cells, brain, microglial cells. We are having different macrophages at different tissues. Coming to the, so till now we have discussed about introduction of immunity, like what is antigen, what is antibody, what is immunity, and what are the cells involved in this immunology. Now we are going to discuss about types of immunity. So we are mainly having two types of immunity. One is innate immunity, the second one is the acquired immunity. Innate immunity is occurred from birth, whereas acquired is formed once we get exposed, then we will get this immunity. This innate immunity is having some different barriers. We'll discuss in the next slide. Whereas acquired is again divided into passive immunity and active immunity. Passive we are having naturally acquired, artificially acquired, whereas active also we are having the same thing, naturally acquired and artificially acquired. So we'll see about innate or natural immunity. We are having non-specific and specific, which will be seen in species, race, individual. So we'll see about these three, or what is the species immunity. So if we see some particular species are susceptible to some diseases, whereas some are resistant to the diseases. For example, frogs are resistant to Bacillus anthracis, whereas toads are susceptible. Coming to the racial immunity, particular race is susceptible, whereas the other races are having the resistance. Example, Negroes of America are more susceptible to tuberculosis than whites. Coming to the individual immunity, the best example we can say, as we have seen the COVID recently. So some are uh, some are susceptible. I mean, like some they fought against the virus and they recovered from the virus, whereas some went for death. So individual immunity depends upon the individuality of that particular immune system of an individual. One exception is an identical twins. Coming to the innate immunity, as I said you, this is a natural or native immunity which will be coming from the birth. So this is the under innate immunity we are having some barriers which will stop the antigen to enter the body. So what are those means? Anatomical barriers, physiological barriers, blood proteins and cytokines. Anatomical barriers. Under anatomical barriers, mucous membrane and skin comes under anatomical barriers. Skin. So the acidic pH will prevent the microbes to grow. So this will protect the body from entering of the microorganisms. Similarly, mucous membrane, so the mucus will entrap the foreign microorganisms. Coming to the physiological values, temperature and pH. Body temperature and fever inhibit the growth of some pathogens, whereas the acidic pH of stomach kills the ingested microorganism, so the microorganism cannot survive. Phagocytic barriers, in what they will do means under endocytosis and phagocytosis they will kill the any pathogen which enter the body. Inflammatory barriers like tissue damage, infection, leakage of vascular fluid, all will protect uh, the body from the pathogens as they will have the antibactericidal activity. Coming to the what are the factors affecting the innate immunity? As A is if there are three things mainly. A is hormones, nutrition. So A is like small children will have less immunity as their immune system does not respond. I mean, does not develop completely, so they are more susceptible to infection. Similarly, the old age people also more susceptible to infection. And there are some hormones which plays an important role for this immunity. Coming to the nutrition, so if we are having enough nutrition, the immunity will be good. Otherwise, we will be having a weak immunity. Coming to the complement system. So what are these complement system is? These are a group of proteins which augment or complement the immune response, which is found in the serum on cell surface. Mostly the complement system, these cells are activated or synthesized in the liver. So under complement system, we are having four steps or four procedures. We can say the one is cell lysis, opsonization, inflammation, immune complex clearance. So the complement pathway, if we see, there is a pathway called classical pathway and alternate pathway. These two pathways finally helps in the formation of the MAC complex. This MAC complex, what it will do means it will cause the lysis of the cell, so the uh, pathogen or the antigen cannot enter into the next system of the body. Coming to the acquired immunity. The acquired or adaptive immunity is the immunity which is developed by the host after exposure to the suitable antigen. So it is having some properties like specificity, diversity, immunologic memory, self and non-self recognition. So under acquired immunity there is active and passive. Active is like induced by natural exposure to pathogen or vaccination. Coming to the passive, it is achieved by transfer of immune products. So it is also having natural acquired and artificially acquired immunities. So what is the difference between active and passive immunity? So the active immunity developed by the host immune system, whereas the passive is a ready-made transfer of immunoglobulin. Active immunity it will be long-lasting, whereas passive immunity is very short-lived. The lag phase will be present, whereas absent is the passive immunity. Booster doses are useful, whereas in passive immunity they are very less effective. Memory component is present, whereas memory component is absent. Negative phase may occur in the active immunity, whereas there will be no negative phase. Active immunity is either natural and artificial. Natural will be seen in any kind of infection, artificial by vaccine. Whereas passive immunity natural is by transfer of immunoglobulin from mother to fetus. Artificial is from artificial, we can say artificially transferred. So coming to the uh, what is this primary immune response, secondary immune response? Primary immune response is nothing but it takes place when the host is attacked by the certain microbe for the first time. Whereas secondary is the same microbe is attacking for the second time, then it is known as secondary immune response. And there are two types of forms, humoral and cell mediated. So what is the difference between primary and secondary immune response? Primary, as I said you, it is the first time exposure of an antigen, whereas secondary second time. Primary will be very slow, sluggish, short-lived, as secondary is very prompt and powerful, and it is prolonged. Lag phase is like 4 to 7 days. Secondary immune response is 1 to 3 days. No negative phase is seen, whereas negative phase is seen. The IgM is the first immunoglobulin which is present in the primary immune response. IgG will be present in the secondary immune response. Antibody affinity will be very low, whereas very high in the secondary immune response. So immunoglobulin production is by naive B cells, whereas secondary is by memory B cells. Humoral immunity refers to the production of secreted antibodies by the plasma cells and antigen specific memory B cells from antigenic stimulation and activation by B cells. These antibody eliminate the antigen by neutralization, opsonization, and complement fixation or activation. Cellular immunity refers to the production of effector cytotoxic T lymphocytes and memory T cells. It will kill the bacteria by killing the cells which are bearing that particular antigen or pathogen. So this is a different procedure for humoral and cellular cell-mediated immunity. What is the difference? So it will mediates by the antibody, whereas cellular is by cytotoxic T lymphocytes. T lymphocytes plays an important role in humoral immunity. T lymphocytes plays an important role, whereas the antibody travel through blood circulation in humoral immunity, whereas in cellular immunity there will be cell-to-cell contact. Primary defense against extracellular pathogens, whereas intracellular pathogens is in cellular immunity. Finally, I will conclude the topic by giving the difference between innate and acquired immunity. Innate immunity takes place in minutes, whereas acquired develops in days. It is present from the birth, whereas acquired after exposure to antigen. Antibody diversity is limited, whereas it is varied. Memory receptors are not present. Memory receptors are present as this memory will recollect, and if the antigen enters, again the body will respond according to that. No particular receptors will be present in immunity. Specific receptors will be present in the acquired immunity. So this is about all. This is all about immunology. Hope you people understood.